Okay, so we're going to talk a little bit about scientific inquiry here. Um, I'll link to a little video on some lab safety that I'm going to have you guys look at first. But let's get right into beginning here. So, living environment. What is the living environment? Well, it's all of us and every other living thing out there, and how they interact with and occupy their place on Earth. Everything works together. Okay? We got a couple different spheres on the planet. The one we're going to focus on is what's called the biosphere. Prefix bio means life. It's where we get the word biology. Even though we don't call this biology class anymore, we call it the living environment because it more encompasses everything that the course talks about, but it still means essentially the same thing. We're talking about all the life on the planet. All right? The way we gather information is through a process called observation. Okay? It's not just looking at something, all right? It's all of our senses interacting with the environment. This is how we gather information, this is how we gather data in order to use that information to make discovery. All right? Once we have that data, we can then make some inferences about it. All right? We interpret it. We use our past knowledge and our past experiences to make an inference about what we're observing out in the natural world. And then we take all that information and we group it. Right? In order to work with information, to work with data, we have to have some kind of way to organize it. And so we create classification systems. Now, maybe all of us in class here, if I were to say, let's classify all the people in this room, we might say male and female. We might say over 5'6", under 5'6". We might say something to do with eye color. But we have to create a system so that we can organize that information, and we base that system on some characteristic that we create. All right? All right, so scientific method, scientific inquiry. We have this information, we have this data that we have collected through our observations. And then we take that information and we try to make an educated guess about what's going on. We call that educated guess a hypothesis. All right? You can see this predicts the cause and effect relationship between two variables, those variables being independent and dependent. And then the key thing is test it. We have to be able to test our hypothesis. If we can't test it, it's not science. And quite often, when we're creating this hypothesis, we often write it as an if-then statement. All right? So, for example, let's say we have a question. Does some fertilizer that we have, Bob's fertilizer, whatever, Magic Grow, whatever we're working with, some fertilizer, we call it fertilizer X. If we use that, does it increase the growth of our houseplants? That's what we want to know. Should we buy this stuff and does it work? In other words, the cause is using the fertilizer. Does it have an effect on the houseplants? If we want to know this, and so we create a hypothesis. Now remember, this might change, or we might find that it's not correct. Well, that's what science is all about. All right? So we might say something like, if fertilizer X is added to the soil of our houseplants, then the plants will grow faster. That's my hypothesis on this one. I'm saying to you, I think if we put this fertilizer into the soil, it's going to make my plants grow faster. That's my educated guess. Based on past experience with fertilizers and with plants, I'm going to make an educated guess and say, if fertilizer X is added to the growth of houseplants, then the plants will grow faster. If-then statement. And then we test to see if I'm right. All right. So I want you to try it here for just a second. So what I want you to do is pause this video and then see if you can create a hypothesis from these 4 questions. So question 1, does refrigeration inhibit bread mold growth? Well, you have to understand what the word inhibit means, okay? Inhibit means slowing it down. So what the question's asking is, if I put bread in the refrigerator, will it slow down the rate that mold grows on that bread? So you have to create an if-then statement. And you might say something like, if bread is refrigerated, then mold growth will be inhibited, if you think it does. If you don't think putting bread in the refrigerator will slow down the growth of the mold, you would say, if bread is refrigerated, mold growth will not be inhibited. All right, so we have an if and we have a then. If bread is refrigerated, then mold growth will be inhibited. That's my hypothesis for that one. And you would do the same thing for the other ones. So again, if you haven't already, go ahead and pause this video. Try to see if you can figure out a hypothesis for questions 2, 3, and 4. All right, hopefully you're back now. You've done it and figured it out. So let's take a look real quick. Question 2, does acid rain affect the germination of dandelion seeds? Germination, when you put seeds in soil, they start to grow, right? Okay, well, if you have acidic rainfall on your soil, is that going to affect how your seeds germinate? And again, depending on what you believe, what your educated guess is, you might say something like, if there is acid rain, then dandelion seeds germination will slow down, or something like that. All right, question 3. Is drug Z, and remember, whatever drug we're talking about, just we're using a letter, okay? Is drug Z effective at treating bacterial infections? And again, you might say something like, if drug Z is used, then bacterial infections will decrease, or something like that. That's my educated guess. Again, I haven't tested it yet, so I don't know, but I'm just guessing. All right. All right, question 4. Does adding compost to the soil of tomatoes make them grow faster? If you don't know what compost is, it's all your food and eggshell waste and all that kind of stuff that gets put outside. Usually have a container or a pile somewhere, and it gets turned and twisted, and over time bacteria in the soil break it down, and pretty soon you have a nice, nutritious, fertile material that you can add to plants and things, and it will feed the plants. And that's the idea behind it. So my hypothesis might be If compost is added to the soil, then tomatoes will grow faster, or something like that. All right, working backwards. What this means is, let's say you're given the hypothesis. I'm not going to spend a lot of time on this, but if you're given the hypothesis, you could work backwards. What was the question? So I know this is a little hard to see because of this thing down here, but it basically says if overweight people take some diet pill every day, then they will lose weight. That's the part you can't read at the bottom. It's covered. I apologize. So if overweight people take diet pill M daily, then they will lose weight. And if you want to work backwards from that, you could simply say your question could be, does taking diet pill affect weight loss, or something like that? All right, moving on. All right, design and conduct a controlled experiment. Once you have your hypothesis, then you have to test it. All right? So you're going to need a couple of setups. One, you've got to have an experimental group. That's where you test whether your hypothesis worked. And the other is called a control group. And the control group is your reference point. It's what you don't do anything to see if there's a difference. All right. So for example, let's use the fertilizer one that we had before. Okay. Does fertilizer X increase the growth of houseplants? Again, our hypothesis was if fertilizer X is added to the soil of houseplants, then the plants will grow faster. All right, so in our controlled experiment, we have the control group, which we put no fertilizer in. So we've got a bunch of different pots with the same plant, right? We want to make sure that our conditions are the same if possible, right? So we've got the control group where we don't use any fertilizer, and we've got the experimental group that we use fertilizer on. So again, we have to have two setups because if they both end up growing the same amount, we can say there was no effect. If the group on the right grows more, then we know our hypothesis was correct. However, on the other side, if the group on the left grows more, that might mean that our fertilizer inhibited the growth of the plants. That's something else we can look at too, right? But we always want to set up a controlled experiment. We always want to have one group that we don't do anything to, or at least we don't change anything, and then the other group where we change one thing. So remember, in both these groups, it's the same soil, the same plants, the same amount of sunlight. Every variable that we can think of, we kept the same. The only thing that was different is in the experimental group, we added fertilizer X because that's the hypothesis that we're testing. All right. And again, it depends on the fertilizer as to the results you get. You might have one fertilizer that makes them grow a lot. You might have another one that doesn't work well on that plant. So that's where you have to kind of test and figure out what works for you. All right, so here's some questions. All right, I'm going to read the question, and then I'm going to have you pause the video for a second and try to answer it, and then we'll move on. All right, so the independent variable is the single factor that the scientist is testing. It is the only factor that the experimental group receives and the control group does not. What was the independent variable in that above experiment that we just did? Remember, it's the thing that changed in the experimental group that did not change in the control group. Right? So what was the independent variable in that experiment? Hopefully, you said fertilizer. That was the single factor that we're testing for. So that's our independent variable. We're controlling that. Okay, we're not putting it in the control group, but we're putting it in the experimental group. Right? All right, question 2. The dependent variable is what will be measured or observed. It's the factor that is affected by the independent variable, the fertilizer. So what are we measuring? What is the dependent variable in that experiment? Hopefully you guys said plant growth. That's the if-then statement. If fertilizer, then plant growth. That's kind of what we're doing, independent and dependent. I mean, that's what we're looking at. All right, once we've conducted our experiments, we're going to make these observations and we're going to collect our data. After setting this up, you must then decide on a type of data you will collect and prepare a data table for recording your observations. All right, so our title, here's how, notice what the title is, okay? Notice the first 3 words, the effect of. We can always create a title for our data collection, our data table, and then from that, extending on that, is our graph that we're going to create, all right? You can always create the title by starting with the effect of. And then you punch in whatever your variables are. Independent and then dependent. So you could say the effect of independent variable on dependent variable. You can do that for every single one that we do. In this instance, it's the effect of fertilizer X on plant growth. Fertilizer X is our independent variable. Plant growth is our dependent variable. All right, and you can see the data table. Okay, we've got height of houseplants in centimeters. On the left side, we've got A, B, C, and D, 4 containers. These are the plants that were grown without fertilizer X. That's our control group. Those are our 4 control plants, A, B, C, and D. On the right, those are our plants grown with fertilizer X. Those are our experimental group, E, F, G, and H. All right, and then now let's get on the left. Week 0. Okay, week 0, that means that's our starting point. That's before we've done anything. Notice they all are the same height. We got 8 plants that are the same height. They're all 10 centimeters. A, B, C, D, E, F, G, and H. They're all 10 centimeters tall. That's our starting. And remember, we got them in the same soil. We've got them the same amount of water, the same sunlight time, everything. Look at 2 weeks later. 2 weeks later, plant A grew 1 centimeter, B grew 2 centimeters, C grew 1, and D grew 1. Now look at E, F, G, and H after 2 weeks of using the fertilizer. E grew 4 centimeters, F grew 2, G grew 2, and H grew 3. So we're starting to see a change. Okay? Starting to see that the experimental group, E, F, G, and H, they're a little bit taller after a couple weeks. And then look at after 4 weeks, 2 more weeks, week 4, and then 2 more weeks, week 6. Look at the final numbers. After 6 weeks, our control group, A, B, C, and D, the ones that I didn't give the fertilizer to, A went up 5 centimeters, B went up 4, C went up 6, and D went up 5. They grew an average of about 5 centimeters. Oops. Plants grown with the fertilizer, look what happened after the 6 weeks. E went up 10, F went up 12, G went up 10, and H went up 8. So they grew an average of about 10 centimeters. Mm-hmm. So our control group, without me adding any additional fertilizer on their own, they grew an average of about 5 centimeters over 6 weeks. Whereas our experimental group, the one that I added this fertilizer to, this nice composting, nutritious material, they grew an average of about 10 centimeters. So they about doubled their growth over the other one. So right now I'm thinking, yep, my hypothesis appears to be true based on the information that I've collected so far. All right, so now we analyze the data and we draw a conclusion from it. When analyzing the data, you simply look to see if there's a cause and effect relationship between the independent and dependent variables. A conclusion is simply a statement that either accepts or refutes. Refutes means it goes against it. It says, no, it didn't happen. Okay, so we're going to base our conclusion and say, is our hypothesis true or is our hypothesis false based on the data we collected? Now, granted, we only did 4 pots in the control group and 4 pots in the experimental group. If we had more data, maybe we had 100 pots in each group, then we would have more data to draw a conclusion from, and we can make a better conclusion. But simply based on the information that we had, we can pretty much say that fertilizer X is probably good for the plants, right? So based on the above, does the data agree or disagree with it? Well, based on what we have, it does agree with my hypothesis. Okay? What is the significance of whether it agrees or disagrees? Well, if it agrees, then we can move on and we can use that fertilizer and try to use it in some of our other plants, test those. If it disagrees, then we might have to go back to the beginning and maybe change our hypothesis. Okay? State a conclusion drawn from the above data. Well, the conclusion would be that adding fertilizer X, or a good conclusion would be adding fertilizer X, whatever it is, to my house plants will make them grow more or faster. Right? How can you make the conclusion more valid? Well, I already said, how can it be more valid is if you have more data. Right? If you take a coin and theoretically there's 50% chance of getting a head, 50% chance of getting a tail. Okay, but if you flip that coin 5 times, you might conceivably get all 5 heads or all 5 tails or some combination. The more times you flip the coin, the closer the percentage will come to 50-50. If you flip it 10 times, you might get, 7 and 3, 6 and 4. You might get 5 and 5. You might get 10 and 0. We don't know. But if you flipped it 100 times or you flipped it 1,000 times, the more times you do that, that's collecting more data, and that's going to make your conclusion more valid. It's going to get you closer to that 50%. And number 4, why might other scientists want to conduct your experiment using different kinds of houseplants? Well, all we did was use one houseplant. That doesn't tell me whether the fertilizer will work on this other houseplant. So once you find out, it appears to work really well on this plant, all right, now I've got this other one. Will it work on that one? Well, I better test that one, and then I got to test that one, and then I got to test this other one. All right, because just because it works on one thing doesn't mean it's going to work on everything else. All right, so you can't really make a statement saying that it's definitely going to work on this other house plant just because it worked on this one that we did the test on. You've got to test everything if you want to know what it's going to do. All right, so let's go back and recap a little bit. What is the question or problem? Do seeds require sunlight to germinate? It's another example, right? So we're going to formulate a hypothesis. Now, notice I didn't write it as an if-then statement in this instance. They don't always have to be written as an if-then statement. You can write them that way. I could have said, if sunlight is given to seeds, they will germinate better. That's one way I can do it. This is just another way to do it that's maybe a little simpler. So here's my hypothesis. Yes, seeds require sunlight to germinate. All right. Design and conduct a controlled experiment. What is the independent variable? All right, well, we've got a control group, right? We're talking about sunlight. So what we're going to do is we're going to have 2 groups. One group of seeds are going to be kept in the dark, and another group of seeds are going to be exposed to sunlight. Now remember, what do you do with seeds? You bury them in the dirt, right? So they're going to be underground, both of them. So light, the amount of sunlight, is our independent variable. right? One group's going to be kept in a cabinet in the dark, and the other one's going to be out on your counter or your windowsill exposed to sunlight every day. All right, so now we're going to make some observations and we're going to collect some data. Oh, don't forget, all of the variables are kept the same. That's important, right? In our control group, all the other variables are kept the same, right? So what is the dependent variable is whether the seeds will germinate. Are they going to germinate in the group that's in the cabinet in the dark? Are they going to germinate in the group that's on the windowsill in the light? That's our dependent variable. That's the result we're looking for. All right. Just like before, did you make a data table? Because we're going to be measuring and looking day after day or week after week or whatever your time frame is. So you got to have something to keep that data in. You got to, all that data you collect, you got to have a place to put it. All right, so we're going to analyze the data. Did the seeds germinate in either or both groups? We're going to draw a conclusion. And if you have ever done this, and you think about the fact that seeds are underground, you bury the seeds in the dirt. So realistically, neither seed is exposed to sunlight because it's underground initially. And if you do this, you'll see that, yes, even the seeds that are in the cabinet, in the dark, they will germinate and start to grow. Now, once they break the surface of the soil, then yes, you're going to need to keep them in sunlight in order for them to continue to grow. If you keep them in the dark, that will inhibit their growth. But we're just talking about germination, the starting process of the seed growing, right? So we're refuting our hypothesis. Look back up. Our number 2. Our hypothesis was seeds require sunlight to germinate, and we just found out under number 5 that they don't. So we're going to refute the hypothesis, and we're going to report our findings for other people to. Now, here in high school, if we do an experiment in the classroom, we're going to kind of do it with each other. We're not going to put it out there into the world for other people to try. But if you were working in an actual laboratory and you did some scientific experiment, you would then report your findings when you're all done in some reputable journal so that other people can see what you did and they can replicate the experiment and see if they get the same results. Right? If they're not getting the same results, then there might be something that you need to change. All right? Stop right there. For today.